So we're going to dive into science panel number 2, and I am thrilled to have some really amazing experts alongside us on the table on the panel with us this morning. But before I do, I want to put it in some context of what we alluded to yesterday. And for those of you who were here on Thursday, you may have caught some of the UMDF sessions that were just simply fantastic. Phil Yeske led a really interesting dialogue around translating science into trials and into therapies. And we had a chance to learn from and hear from other patient advocacy group leaders who maybe have further along on some of the development of new therapeutics. And two things really stood out for me from that conversation Thursday morning that I think tie into some of the things we're going to be talking about. One, patient communities are the key to supporting the learnings of how do we not only live with, but in many cases thrive with rare diseases such as LHON. And if I just saw the energy this morning and all day yesterday and the sharing among all of you on tactics, techniques, coping mechanisms, and ways in which we're finding not only how to live with, but to actually flex various superpowers, you guys are an incredible example of this type of a patient community really supporting one another. And it's that powerful community that I think really sets the stage for the second, for what they were really talking about, which is how do you not only support one another, but how do we actually plug into this ecosystem around us of researchers, of clinicians, of industry partners, and dare we say, even talking with and informing regulators around the world on what measures of success could actually look like. Two big phrases that stood out for me. One, hope is ineffective without action. I'll say that again. Hope is ineffective without action. And this is an action-oriented group, ladies. You're going to get to know this is an action-oriented group that likes to lean in. And as I was kind of reflecting on that, the idea of how do we really get involved in each person's journey when you're ready? And I hope this morning was a good example of how many different opportunities there are to get involved, to act, and to be a part of leading this community forward. So with that, I think we just have to start by giving a quick round of applause to our incredible conference organizers, to Jessica, to Maria, to Alyssa, to Brooke, to the entire team that pulled this morning's coffee and conversations together. You guys are amazing. And I think within this broader context of the community update yesterday, it was just so clear how many efforts are underway. And what I hope you also took away is that there's a real strategic roadmap that's developing here of how these things actually work together to band our community together and to connect with that ecosystem and advance the science as well. So it's my pleasure to introduce my friend and our partner at the Milken Institute's Spark team, Ms. Dr. Caitlyn Barrett. And Caitlyn's going to introduce our amazing mitochondrial experts who are with us as well. But Caitlyn comes to us with an incredible background as a researcher. As I mentioned yesterday, she works with Milken Institute and specifically leads all of their rare disease portfolio of programs around the world. So Caitlyn, I'd love for you to maybe just kick us off. Give us a little background on what informs your perspective in this whole journey and maybe a little of the Milken Institute as well.
Absolutely, Chris. So I already see an issue here with me and Anne sharing. We'll be fine. Thank you so much for the introduction, Chris. And I really want to thank the organizers. It is such an honor to be here. And I have really, really enjoyed my experience starting to get to know this community. The Science Philanthropy Accelerator for Research and Collaboration, SPARC, is the organization that has been partnering with LHON Collective. And we're really thinking about our work is centered on figuring out how we can create impact and accelerated movement within the biomedical ecosystem. And the way that we do that, not one person can be an expert in any disease, is that we actually leverage the experts. We talk to foundations, we talk to regulatory, we talk to researchers like Dr. Falk and Dr. Chiaramello, and we try to better understand what the research landscape looks like over the past 30 years, for example, of LHON research. We see where there have been successes. We see where there have been challenges that can be overcome. And really, we link that with what the funding landscape looks like. Why have we seen stagnation in certain areas? Why have we seen boosts in certain areas? And where can really strategic philanthropic funding come in and make the difference within a research space? So what we have done, I'm going to try and be brief because it's much more important to hear from the others at the table. But what we've done is we've spoken with over 70 experts, patient, advocate, researchers, clinicians, to better understand LHON. We also had an expert convening, and both Dr. Falk and Dr. Chiaramello were actually in attendance at that meeting, so they'll be able to share a little bit more. But we brought almost 30 individuals together from the LHON space, from the mitochondrial space, from neurodegenerative, from optic neuropathies. And we pulled them all together and we said, if we are going to focus on LHON, what is the path to make real progress? And we will talk a little bit about what came out of that. But what I can tell you right now is that there is a clear path forward. And hope and action can really align well when we think about the strategy behind supporting research and building that development pipeline. Without further ado, I would really like to welcome the experts on the stage. We have, to my direct left, Dr. Ann Chiaramello. She's the professor of anatomy and cell biology and the director of the Mitoepigen program at George Washington School of Medicine. And Marni Falk is a professor of pediatrics at the Perelman School of Medicine at the University of Pennsylvania and the executive director of the Mitochondrial Medicine Frontier Program. Thank you both so much for being here. So I mentioned in that very brief introduction that one of the things we did was pull together experts in a range of different areas. And we talked about what do we know about LHON? What do we need to know about LHON? And how do we cross that bridge? I would love to hear from you first and foremost, and we'll start with Ann. What do you think the utility is of bringing all of these different people together, and where did you find the most benefit from that meeting?
Well, now I feel like I'm coming back home because, I mean, I started with the LHON group in 2016. I met Lisa. Everyone knew Lisa, but it was the first time I met her. And at the time I met her, I was working on another mitochondrial disease called MELAS. It has the same symptom, similar symptoms, and at least the same molecular charges than LHON. And she said, why don't you come cross over and come to the LHON group? I said, well, let me go on the wild side then. I mean, how can you say no to Lisa? That was in 2016. And, 8 years later, I'm still here and I enjoy it. So I think that I've known a lot of you and I feel like every year I'm coming back home. So thank you for always making me feel welcome over those years. And thank you, Lisa, for really, really opening it up because she was instrumental in actually creating this new way of thinking for clinical trial to deal with more than one disease at a time. And that's called basket clinical trial. And now the FDA approved the first basket clinical trial on 2 mitochondrial diseases in the United States. So I think that I owe a lot to Lisa. So thank you. And of course, more recently, I met Chris Marsh, Melinda, and Caitlyn, because we're neighbors.
Yes. We had coffee. That was our first meeting.
Yeah.
In person.
Right, on campus. Can you believe that? So I said, oh, I didn't know you existed here. I mean, so it's a very small community. So there's something really nice to reach out, but you have to reach out to the right people. And I always felt like I was somehow guided because I stumbled on the right people. and the right community. So I think for me, to have worked with the patient advocacy group like LHON Collective now, it's called, it has been instrumental. Not only it puts the research we're doing in the lab, the translational and clinical research in perspective, because we don't lose the perspective that there are patients waiting for some help here. So it guides us, and Martine is in the audience. She's a lead research scientist dealing with all the patient cells. So she knows all the patients quite well now, intimately well. I mean, and it keeps us focused on you and not just on anything else. Like, because, you know, scientists have a tendency just to sniff around and, you know, like you go on a tangent, but to work with patient advocacy groups keeps you focused, because that's you. We want you just to get better and to provide some help on the way. So I'm going to pass along to Dr. Falk.
Yeah, and actually, let's pause 1 second. And because the perspective that you brought to that meeting was aligned with the patient community. What did you see when we were at that meeting and we brought all of those people together? Was that focus still there? And did you feel like that was something that you could add to the equation?
I think so. What it brought to light is the fact that, like, there's not a single answer there's not a silver bullet. It's going to be multiple answers coming from multiple angles. So it was really good for me just to hear from angles that I would not consider or I would not be in contact with. And then it made me realize that because it's such a heterogeneous disease that we're never going to find just a silver bullet for everyone. So there's a need just to build the scientific community and to hear from different specialists.
That's perfect. Thank you so much. I agree completely. Marni, what was the benefit of pulling all of those people together in your mind?
To me, that was an enormously beneficial meeting. Of course, getting to meet all of these wonderful organizers. It's incredibly important to have sort of the voice and the articulate the need of the community. So hearing that was really important. My history is I'm a geneticist. So I think about, the gene and what symptoms the gene cause. And I've noticed a lot of doctors like to make categories about how to limit that. This affects the eye. We're going to call that LHON. This affects the heart. We're going to call that cardiomyopathy. This affects the brain. But I don't think the body knows that to the same extent. So I've seen people with the 11.778 mutation who have multiple sclerosis, or who have neuropsychiatric serious problems, or who have cardiomyopathy. When I started out my career at Children's Hospital, I was running the eye genetics clinic. So it's funny how it all circles back around. And I would examine people in the ophthalmology chair, which is an awkward place. Many of you probably know to do a full exam, right? You're sitting up. And what I noticed, it was a joint clinic with myself and an ophthalmologist, wonderful retinal specialist. They didn't look beyond the eye. So when they were telling me LHON affects the eye, they didn't ask if there were other problems. So I started seeing LHON patients in the mitochondrial clinic, where we think about the genes and the mitochondria. And I started noticing there's a lot of other conditions if you ask: the gastrointestinal symptoms, the fatigue, the exercise. So I think what was so amazing about that meeting is that it was centralized on the ophthalmologist. And I think I had sort of let it go. The ophthalmologist got this. They're going to cure LHON. But not if they're only treating the eye, right? And so I think that was one of the big conversations was, yes, it's incredibly important to treat the eye, and how do you prevent people from getting problems, with their vision, and how do you sustain and restore vision? But how do you actually find out what's important to the families? And I think that's what, how much improvement is good enough, I think if you ask somebody, they would say 100% would be ideal. That would be great. And so, how do you match that with sort of what clinical trials are able to do? And so I think that's what I took away from the meeting, is that you can't just leave this to one type of doctor and expect everything to be fixed. You need to bring all the voices to the table. What are the concerns? What are the needs? And then what are the treatments? And what is the degree of effect that you want to see? We don't just want to have an approval that does something marginal, right? We want to have an approval or a series of approvals that do something profound, right? That really improve people's well-being. And I think that's when we realize we can't just leave it to the eye doctors in the eye room. Again, no offense, I love them all, but you know, their job is to look at the eye, but the rest of us can't give up our role in it, right? Which is to look at the whole person.
I'd like to echo and expand a little bit on what Dr. Marni mentioned, because it's really important. It opened up a dialogue. So that's what you did. So thank you for organizing this retreat, because it was wonderful. Like we all talk different languages, but we managed just to find like common language. Yeah. So you're right. They're great neuro-ophthalmologists. We don't criticize them, but
They're wonderful. Yeah.
they're wonderful. We love them. But I mean, it's not enough for the patient. So the dialogue was really important. So dialogue with the community is one thing, but dialogue among us, scientists and physicians, you know, that is super critical.
I think that a really important point that's come up in this conversation already, and I'm already going to go off script, so excuse me for that, is the fact that LHON, we focus a lot on the eye. We think a lot about the eye. But as we left that meeting, one of the key LHON experts, neuro-ophthalmologist, stood up and he said, I know the three things that we need to do moving forward. And that is actually turned into, with all of the detail that we pulled together, what's called an opportunity map. And one of the very first things that we heard was, we need to understand the patients. We need to understand their mitochondria. We need to understand not just the affected patients, but the carriers and what their mitochondria are doing. So one of the very first steps, and I know we had the LHON data collection table up here. The very first step is starting to really understand the patient experience and expand beyond the eye and look at the other issues that may be arising and understand how what we call the genotype phenotype connection. So what is the genetic change that we're seeing causing downstream in the actual individual? So I would love to just get your perspective on the importance of having data, patient data, clinical data, and maybe even biosamples. I know in your work, and you're already using some of these things, and I'd love to hear more about how important that is for your work.
Sure. So we in the lab, we work only with human-derived cells, so from patient-derived fibroblasts. So those are skin cells. So from a skin biopsy, we derive those fibroblasts and then we expand them, and then we do multiple studies on that, genetic studies, genomic studies, mitochondrial assays. So we know, we define your mitochondrial signature, your personal mitochondrial signature, and we map that with metabolomics and proteomics. So it's kind of like a very comprehensive approach, and it has potential later on just using the induced pluripotent stem cells, the iPSC, just to lead to neurons, cardiomyocytes, like Dr. Falk mentioned, like all the different symptoms. So it's kind of, we lay the foundation. And it also makes it, make us realize that it's multi-layered. You just have the what we call the LHON mutation or pathogenic variant, but you got to go beyond that. You got just to look at what's the genetic modifiers for each one of them, and maybe that will help us understand why we have carriers that remain asymptomatic and for a very long time, and we like just in a way. That's what we were talking the other day. How do we prevent them from converting to symptomatic? So. Marni, you mentioned that, you know, it would be good just to have like the natural history really well done, so that maybe we can detect the little target signs to avoid these conversions, if you want to support on that.
Yeah, no, I think collecting the clinical real life data is incredibly important. Like you said, you know, there's been some evidence that smoking and alcohol are not ideal, that it pushes conversion. But we've also known that we kind of thought more people converted than they do. So genetic testing is really important, because you might only be looking at the symptomatic patients. And so once you understand who in the family is actually a carrier, you can really start to learn, well, what symptoms do they have? Maybe they don't have vision symptoms. Maybe they have migraines, or maybe there's something else that we need to know about. And then, we were doing a study with a drug company, and we realized a lot of people weren't qualifying because there was a lot of marijuana use. Well, we never even asked if people did marijuana. So how do we know if it was a risk factor, right, for their disease? So I think there's so much in people's lives that are probably influencing their health, their diet, their level of exercise. Exercise has been the only proven therapy for mitochondrial disease. We don't know how it affects the eye specifically, right? But we know that it helps you make more mitochondria. We know that in LHON, people who have more mitochondria in their cells have less likelihood of losing their vision. So would exercise be a factor, to prevent conversion? So I think there's a lot of opportunities both to understand what lifestyle things can be done, what counseling can be done. If only we used to think, a much larger proportion of men and women converted. If let's say only 15% of people are converting, well, what can we learn from their family members? And in every mito disease, those who exercise are usually healthier than those in their family who don't. That said, if you have a heart issue, we probably need to know that before we blanketly tell you to go exercise, because maybe we're trading one benefit for another problem. So to put it mildly, I think everybody's unique, but there's probably some common knowledge we can gain about how to understand the course of the disease, the progression, the resolution, what people themselves are doing. Maybe there's diets that are more enriched in antioxidants or more enriched in certain types of food, that are protective. So I think there's just so much to be learned from the actual families. And then the samples are incredibly important. I started collecting cell lines and samples a long time ago, 2008. So what's that, 16 years ago? And now we have hundreds and hundreds of samples, and which is amazing because now you can turn them into stem cells and retinal ganglion cells and fibroblast cells. And we could do immune studies and look at the interaction, but without samples, all you have is ideas. You can't test them. And so we also really like human samples because even if we're studying another model, an animal model, let's say, and let's say we have a drug that's very promising, but what if in this particular disease, there's some risk, there's some toxicity? It would be really nice to have one day a test where you can say, Dr. Chiaramello has a therapy, but let's make sure that in your cells, it doesn't kill them. That would be good, right? Like looking for no toxicity, because we also know that in the setting of mito disease, the cells kind of adapted. And sometimes those adaptations make the individual more susceptible to medication problems, right? So even if something works great, in a model, it would be really great to have the human samples to confirm. So from a genetic level, from a treatment, verification or validation, from really understanding the disease, and even something as simple as, is a debenone good in different mutations? Well, it'd be really great to test different mutations, and say, this cell line looks actually sicker. This cell line actually looks better. So to be able to stratify. So I think you can't underestimate the importance of samples, and the world's gotten much more sophisticated at creating them and using them wisely and banking them and sharing them. So I think these are, you don't have to go through a skin biopsy with every doctor. I think these are cells that can be immortalized or be used for a long, long time if they're handled properly and shared, so that you could also compare apples, to oranges, so to speak, right? Like if researcher A has a treatment and researcher B has a treatment, well, if there was a standard set of cells, you can actually compare what was the effect here versus here. If everybody's always using different cells, sometimes it's confusing.
Caitlyn, if I could jump in just for a second. This is exactly why we had a discussion yesterday about the data collection program and why I was so pleased to see so many of you come over and talk with Melinda about how do we bring what you're experiencing into a standardized and structured set of data that can then inform some of these insights. And the fact that we can do that globally now actually certainly helps. But we personally, as a family, went through with Marni and her team the whole skin sampling, and this idea of actually not only those who are affected, but carriers, but even, what am I, wild type?
You're just a healthy control.
Like
from a mitoDNA perspective.
healthy, questionable, but the idea of being able to have something that can be compared across and within a family and be able to actually look at some of these things as the knowledge base
Right.
builds.
There's
right?
something called a cybrid. I'm sure you've heard this crazy term. It's called the trans mitochondrial cybrid line. Dr. Wallace and Dr. King, they came up with this concept decades ago, but it's the way to prove that a mitochondrial DNA variant is like the cause of the problem for the health of the cell or the individual. You take a common nucleus and you get rid of their mitochondria in the cell, and you swap in either the patient's mitochondrial DNA genome or, like, let's say their mom's. Let's say their mom doesn't carry it. So then you compare their mom, which is identical to theirs, except for their mutation. So maybe one has 100% of the A, one has 100% of the G, and you compare, right? That's been the gold standard in the field, because you're saying whatever difference you're seeing, it's because of this one mutation. That's done from a blood sample. We do that from platelets. So that's the type of thing that's like the gold standard biology. And that doesn't even hurt. That's a blood sample. And then once you create it, they basically can live forever and be shared.
So Caitlin, I'll pass it back to you, but I want you to picture what Caitlin described of 30 brilliant researchers and scientists from around the world in a horseshoe room, sitting in a hotel in Lisbon, Portugal. And yes, everybody got to get out in the evening, but for basically 2 full days. And listening to Anne and Marni bring such thoughtful questions to the equation and help challenge the thinking so that we really do bring together 3 decades worth of knowledge up to this point and inform how we go forward. It was really awesome to have you facilitate that, Caitlin.
It was a pleasure. And Anne, did you?
Yeah, I'd like to circle back to what Dr. Falk and Chris just said. And if I would unpack a little bit what Dr. Falk said, because I mean, it's lots of information. So first of all, I think that what the bottom line, what the take-home message I took from the Lisbon retreat is that what will benefit most of the patients is a holistic approach. So it was a holistic approach in terms of scientists and clinicians, because you put us all in that room and we were forced just to talk. I mean, look, you live in Philadelphia, I live in Washington, D.C., and how often do we talk?
We had just met. Yeah.
No, that's right. And look, we're back again. So you guys are matchmakers. So the holistic approach was really good, but at the same time, that holistic pattern or model can be applied to science. Like you said, we need a holistic scientific approach. No, it's just not just going to be one approach that's going to make it. It's going to be several ones, and we have to tackle that with different specialists, genetics, and mitochondrial specialists, and proteomics chemists. I'm going to forget chemists here.
Yeah.
But
Regulatory.
the most important one is the new one, the AI, because AI will be our friend and your friend, because it will be able AI will be able to look at all the data from different angles that your brain, my brain together will not be as efficient as that. And then we are going just to lead AI just to solve a little bit of mystery. So I think AI, I'm not a big fan of AI, but I have just to become one, because I think that it's going to help us and help the community.
Some of the accessibility tools that I've been seeing are already engaging it. So why are we far behind? I think that Dr. Falk and Dr. Chiaramello mentioned something in passing that I think was the next important step. So we talked about patient data. There is another piece. And when you think about research, you have to have tools. And you wouldn't take, unless you're me, a hammer and try to change a tire. I had a lug nut problem, to be fair. What you need is a tool that matches your needs. And we've heard about cybrids. We've heard about stem cells. There are things called organoids, which is actually taking a stem cell and growing out a cell population with a lot of variety. You can do a brain organoid. We haven't gotten into animals yet, but I definitely want to talk about that. It's really critical that there's a toolbox. We have to answer a specific question, and instead of saying, This hammer is going to be used to change this tire and to redo this plumbing and to do some electrical work. What we need to do is have a toolbox that's going to say, okay, we need an animal model that we can rapidly test therapeutics in. Zebrafish, we'll get into that. We need an animal model that's going to be able to tell us a little bit more about the safety of a therapeutic. Maybe a mouse. We need a cell model that is going to help us learn the biology of the disease. So I'd like to step back. We have all of the experts we need, but we also have to have a variety of tools. And I would love to hear more about how you guys use tools as you're doing research on mitochondrial diseases and LHON. And I'd love to start with Marni, because I think that there's some really cool things you can share with us.
Yeah. So we came up with this idea of therapeutic cross training. So they're making me run a marathon. Don't ask. But when you're preparing for a race, you probably also want to swim. You also want to walk. You also want to bike, right? You want to build up different muscles. You want to build up different skills. You probably want to pray. There's like a whole lot, you want to do to get through that race. I think it's the same thing when you're developing a therapy for a person. You don't want to play in a person, right? You want to find a model to do screens in where you can fail, right? And there's no serious consequences, but you want to fail quickly. So we use a whole series of models. We use microscopic animals, actually, that are really small and inexpensive, something called C. elegans, where the biology is really well conserved. These are worms that were developed as a model by Dr. Brenner back in the 70s for neurobiology, so neuroscience, that goes from 1 cell to 1,000 cells in 3 days. They make hundreds of animals each that are genetically identical. You can imagine you have no limitation of genetic material. You can manipulate them. You can learn. We can screen thousands, hundreds of thousands of compounds incredibly quickly, right? And what's remarkable, we can find dose. We can find where's the toxic dose? Where's the therapeutic dose? We could test combinations. So we do this now for many mitochondrial diseases, and it all started with a complex 1 disease. So my expertise, my interest has always been in complex 1. That's the genetic part of the electron transport chain that has 45 subunits where which is fascinating, all of the LHON mutations hit complex 1, right? And so we can model energy impairment in complex 1. It's like a Lego house, right? 7 of the subunits in the core are from the mitochondrial DNA. The rest all around it are subunits from the nuclear DNA. So why does LHON primarily show up with some of the mtDNA subunits? ND1, ND4, ND6 are the common, but there's others. And I actually have seen lots of other people who have optic nerve problems with other complex 1 genes. So there's probably a lot to learn here. And we don't really know yet. And this is the big mystery in mitochondrial biology. Why do you predominantly affect, the retinal cells, the optic nerve with these mutations, whereas other mutations affect the basal ganglia or the cerebellum or the heart or the liver. So that tissue selectivity, we don't really understand. We also know it's not very strict. The body can do whatever it wants. And that's why we don't really trust it, right? We look and we make sure, is there a heart problem? Is there a liver problem? And so on. But back to the question about models, you can find therapies, you can investigate disease, you can knock out genes, prove the pathways that are involved, come up with new ideas, have a playground to play in. But worms have neurons and cells, they don't have eyes. So then we moved up to zebrafish. Zebrafish, you can buy at the fish store. We don't buy those, but you could. They start out really, really small, a couple millimeters big, but they're a fully formed animal in 1 day. So it goes from an egg to a fully formed animal with a beating heart, swimming around in 24 hours. That's my type of speed, right? And then each animal is clear. They're transparent for their first week. We give them a chemical so you don't see their zebra stripes. Yeah, they're clear. So you can label things like the eye and really look at how it's developing, and you can study it. And it's got so much of the conserved mitochondria and genes, including the LHON related genes and now the big kicker is that the technology has finally caught up with the ability to manipulate and create models in the mitochondrial DNA. So Dr. Ecker here at the meeting has created the first LHON zebrafish, which they're characterizing now. And this has been a really big problem in the field. There have been no models that are like the people with the mutations in the mitochondrial DNA at what we call heteroplasmic levels, meaning not necessarily every mitochondrial DNA has the mutation, just your proportion do, and it could be different in your blood or your urine, and so it could be hard to guess what it is in your eye. So now that there's a model, well, now we have like a zebrafish ophthalmologist who can do the same tests that you guys get, you know, the OCT and the retinal exam and the electrophysiology, and we can start to see what's their degree of problems, how does it change over their lifespan, and if we stress them, right? If we give them, you know, ethanol, if we give them, you know, virus, if we give them, you know, poisons, what happens, right? So we can actually bring out the problems and start to see the combinations that do it, and we can prevent it. So now the reason I say cross training, you can start in one model and then prove it in the next. So you might see it at like a whole animal survival level, move on to a vision level. And then there are some mouse models of LHON, not all of them, and it's very hard. The body doesn't want to keep the mutation in these animals, so it kicks it out. So you can create it, but then you can't breed it to the next generation, so you can't share it. So that's why we have to be creative and think about different models. And there's other ones out there too. Some people like flies, like the ones that come on your bananas, those are Drosophila. So we can use a lot of things and be very creative. But the problem in LHON that came up at the meeting, and I think everybody agrees, a lot of the advances that were made were guesses. We think this should work. And they jumped really quickly to people. And even some of the technical tests you might want to do, right? Like, do you inject here or here? Do you go in the middle? Do you go in the back? Do you go in the side? They didn't have a model to play in. So they had to take their best guess. So now we all hoped it would work, right? But maybe the reason some things didn't work is technical, right? They didn't optimize the technique because they didn't have the toolbox, right, to really play and manipulate. So I think that's what came out of the meeting, is that we don't want to limit ourselves, right, to guessing. We don't want to limit ourselves to just one model. We'd like to now that genetics has gotten so advanced, stuff like CRISPR and tail-based gene editing and just really cool stuff that's like making it really unbelievable time in science to be in the laboratory. So now we can, 10 years, when I started, 20 years ago, you couldn't do this, right? You had to take what existed. You couldn't create. The fact that you can create models and then share them and characterize them, and you can look at their heart and you can look at their eyes and you can look at their movement and their function, and it's unbelievable, right? And so now it's almost like we're obligated to, right? Like we need now to test, and these things can also be done fast. The zebrafish, every single week when you mate them, you make 100 offspring per pair. So you could have thousands of animals to test every week, right? So these are things that really enhance our toolbox. Again, that's just one model. Sharing fish, sharing cells, and then having kind of like the community say, okay, researcher X, tell me, how did your treatment perform in these models? Then you'll understand and say, hey, wait, you got a 50% improvement, but look at this guy. This improvement got a 90% improvement. Oh, but you caused this problem. Hey, they didn't cause that problem, so that you could really start to have kind of like a catalog, right? And be like, okay, here are the models. Here's what we saw. And this is really what other communities that have been successful have done. Like we just heard a lecture this morning about spinal muscular atrophy, right? There was like no therapy. It was the number one lethal genetic disease. And now there's 3 in the last 2 years. And it all came from having models where they said, oh, this is the cell type where the problem is. Oh, this is the problem that we're seeing. Oh, this is the opportunity at the protein level. And then they really went after it. And so once they had the models, the gene therapy went from non-existent to, I think 2011, they thought about it, and 2022 it was approved. So I think, again, models really allow you to fail and reiterate and innovate and identify the best path forward much quicker than you ever could in people.
Thank you so much, Marni. I think that one of the things that we heard at the meeting, in addition to the need for models, and you started to talk about this with gene therapy and access, and there is so much that we don't know We had a whole session at that meeting. What are our assumptions and how do we test them? And I would love to know, Anne, from your perspective as someone that thinks about LHON plus, what were the things that you came out at the meeting for you that you thought were really important to capture?
To really capture, to highlight the plus. Because I mean, I realized that I was kind of like alone in that category. So I think that, and after talking with many of you, I decided, no, I'm the right category. This is good. I mean, I'm ahead of the curve here. The LHON plus does exist, and because it's a spectrum, you know, it's just like in life, you have like two opposite pole side. Here we have the LHON, and the other end of the spectrum we have LHON MS-like. But in between, there's lots of patients with different symptoms, some of them overlapping, some of them not. And that's where, you know, what Marni said about the systems or the different cellular and models systems, that actually is really useful to then to move on to the patient level, to stratify the patient, because there's a need for stratification. And I realized at a conference in Lisbon that actually we need to unpack the middle of the pack.
Really appreciate that perspective. I think I'd like to step back because one of the things that Chris mentioned at the beginning is that we really wanted to build a strategy. And I know that your scientific sessions have been really well done earlier in this meeting, so I'm probably not telling anyone anything new. But a lot has been dedicated to clinical trials in gene therapy and LHON. And what we all need to understand is that even a failure in a clinical trial isn't the end of the world. 90% of clinical trials fail. Most of them do it at the point of assessing efficacy. And I think that there's hope in building an understanding of the disease so that we have multiple shots on goal. Let's think about other ways that we can develop therapeutics. Let's understand the biology better so that the next therapeutic that goes into a clinical trial has a greater opportunity to actually be successful. And I think that that's one of the things that, you know, Chris alluded to, we've kind of talked around, but this idea of how a clinical trial is started, how it is designed, is it a basket trial, how many patients are going to be pulled in, and ultimately, what are the endpoints that we're looking for? What is the measure of success? So I think that one of the things that we've seen is we're here right now, we partner with LHON Collective to think about the multiple shots on goal and to think about how the next therapy that will move forward is going to be more successful, and it involves understanding the biology of the disease. It involves getting the patients involved earlier and having them talk to regulatory and say, this is a meaningful outcome to me. And it involves being really thoughtful about what we are funding in terms of what moves into clinical trials for a rare disease population. I think about this a lot in rare diseases. There are only so many people. How many clinical trials can you possibly run? So we have to be, as an ecosystem, very thoughtful. One of the things that we've proposed, not only in terms of what needs to be done in LHON, but it's the mechanism of how it needs to be done. And that is collaborative networks. And Marni mentioned this. I think it's really critical that if we build models, they are done so in a consortium. You guys all collaborate and talk to each other and work together. Scientists need to do the same thing. So I think I've hit quite a few different things, but I'd love to back up and just talk about what a consortium looks like, what collaborative fund or collaborative research looks like, and how it can make the research process better.
I think by providing like a forum just to interact. And I think Marni was a leader in that because you did a MitoMap. So that was the very first.
Well, I didn't do MitoMap. That was Doug. I did MCDR. I'll explain that after. Yeah.
I still give you credit. Okay. So because the MitoMap is really a cool tool on the website. I mean, where you can enter any numbers on the mitochondrial genome, and it'll tell you whether there's mutation that been assigned with that position in the mitochondrial genome, and then all the list of references and all that. So it's very easy just to maneuver, and it's informative not only for physicians and scientists, but for everyone. And then you expand it with building all those zero efficiency elegance. And but so that's why, I mean, it's really good for me just to know what you're doing, and it's really good for you to know what I'm doing, because then we can combine efforts into that.
Yeah, I think consortia are incredibly important. I think, for example, the United Mito Disease Foundation brought together different family groups back in the 90s. A lot of you probably know the history. And that was a big moment, right? United, right? Like we all have different manifestations, but we're all going to move together. There are people here who will qualify for other mito disease trials. The inclusion criteria usually means you have it mostly now that you have a gene disorder that affects the mitochondria, and you have symptoms in whatever is being tested. So if there's a trial for myopathy, many of them will allow people with the 11778 mutation or the 3460 or whichever mutation you have to enter if you have the problems, right? You have to have a certain problem to be in a trial, right? You have, unless you're doing a trial, let's say on carriers, then you might have no problems per se, you may be pre-symptomatic, and then they want to look at the rate of conversion. Some trials are intervention trials, some trials are observation trials, some trials are diagnostic trials. I think one of the best things that's happened in the last 27 years is this clinical research pavilion that we have here that you guys all know about and probably participated in. I thought it was like truly rocket science. It's very hard to do a trial where you enroll patients. Here last year we enrolled 70 patients in a day. We're lucky normally if you enroll 70 in a year, right? So talk about accelerating, right? The knowledge and the learning. And this has been showing in group after group that the FDA takes this data very seriously and uses this data to then compare, here's baseline historically to some trial that's ongoing. And we just heard the example of two different, the Barsh syndrome and the Friedreich's ataxia syndrome, where that data collected at meetings allowed for a drug ultimately to be improved. But as an example, like we have a family advisory council at our institution from the mitochondrial medicine patients. We're very glad the Marshes participate and share, perspectives of the LHON community. But a few years back, they looked at us and they said, how are you going to develop therapies? You can't look at somebody and even know how their mitochondria are functioning, right? Nobody knows what we have, right? We're silent, we're functional. People have to, it's not like, there's an obvious problem. So we said, oh, you're right. We don't have great diagnostics. So now we got the UMDF, just to show you how it's all related, lobbied Congress to get mitochondrial disease as an approvable topic for funding congressionally mandated disease research. There is more money in the Department of Defense for research than there is in the NIH. This was a major win. It's been about 9 years. Us and others applied for these grants, and we unlocked truly like a $12 million grant to study diagnostics in mitochondria. And that's what's happening here. We're looking for a breath signature. And what's remarkable, the reason I'm telling you that is because we started in the mouse model of LHON, and there is a breath signature we're starting to see of LHON in mice. Well, that'd be super cool, right? To know if that translates to people. And so there's a study where it's not limited to LHON. We're, of course, interested in LHON, for obvious reasons, because it's a primary mitochondrial disorder. And if we can learn that, well, now maybe that signature could be used to identify carriers or to identify, people who may be at a more severe spectrum. So we're still at the beginning. It's not the only outcome measure, but it just goes to show that the community approach, the idea about a basket, that you're looking at people with different types of mitochondrial disease, where LHON is a very important part, but you can learn from the other phenotypes and they can learn from you. And I think, again, most drug companies want to develop therapies for LHON. I don't know if you know that or not, but that is like, I'm on a lot of boards. That is the #1 disease that drug companies are like, oh, it's so easy, right? It's just the eye, right? It's just 3 genes, right? There's probably over 500 genes that cause mito disease. So there is nobody who doesn't have LHON on their drug development list, but then they say, oh, it's all figured out already, right? There's a gene therapy, right? Like we shouldn't get involved, right? And that's where the patient advocacy voice is so huge, because there isn't a gene therapy yet. There's needs for the community to articulate what they want that gene therapy to do. There's need for models to test it better. And there's so many opportunities. Like I can treat complex 1 disease animals in the laboratory many different ways and make them live longer and make them function better. All right. And but how do you translate this to the different problems? So that comes back to the clinical research. If I have a breath test, if I have a walking test, if I have a vision test, we were doing pupillometry. I think some of you probably know that too. And we're developing new retinal tests. So if you can really ask the families how they're doing, the affected people, the carriers, if you can really understand the severity and sort of the impact on their lives, and you can have something to objectively show it, that's how you get to approvals really quickly. And then in the background, you're keeping the pipeline coming of different therapies, because I think of therapy as an arsenal, right? Like you would never expect to treat all cancer with one therapy, would you? Never, right? You'd expect to have an arsenal of therapies and your doctors to have choices. Miter disease is at the beginning. We are going to have an arsenal of therapies, and some of them are going to work for people with certain symptoms. Some of them are going to work for people with certain genetic causes, right? And we need to understand how to layer them together. So that's why I think this consortium is so important. And the learnings, like Anne said, what have you done? What have you seen? It is just as important to share what doesn't work as what does. We don't want Anne wasting her time doing something I already know doesn't work, right? We want to make sure she knows that so she can focus on where the money is, right? Like where the likely success is. And I think in rare disease, there's just not enough of anything, right? There's not enough patients. There's not enough experts. There's not enough models. And so it's our responsibility, to be very sort of careful and also collaborative so that we share what we have to move things forward as fast as possible.
I like to add, like, if I were a patient, I would say, but what can you do for me now? That's a burning question. So that's why, gene therapy is great. I'm all for it, but it's long-term. We're not going just to have something very quick. So that's why we took a different approach in a lab. First of all, we took a mutation agnostic approach, meaning like it's independent of the mutation you have. It's not 11778, it's not 3460. It's not like that. Because I realized that there's so much clinical and genetic heterogeneity. So we went back to the basics and decided, okay, what is a common denominator here? How can we help the patients rather immediately? And we decided just to go a different route than gene therapy, because first of all, there are a lot of people doing that. And so, but I kind of like took the perspective of the patient. What is missing to the patient right now is energy. How can we bring them energy? How can we deliver some energy immediately, independent of their mutation and their genetic background? So we designed like a very simple way just to basically coerce mitochondria, I have a fly here.
It's the banana fly. It's the banana fly.
I'm not a Drosophila researcher, just so you know. I can't stand them. Not my fault.
Okay. So basically, what was I saying?
You were going the non-gene therapy route.
No, I'm non-gene therapy. No. I was saying like, we designed a strategy to coerce your mitochondria, to fool them, to say, okay, because nature always built a back door, we have to find the back door. And we think we found the back door to circumvent your complex 1 deficiency. And by doing that, we decide like, okay, not only we're going to find the back door and open that back door, but we'll make sure that it's going to stay open so that you keep producing ATP, meaning energy, because the bottom line is a lot of patients, they complain about chronic energy exhaustion. And that's not fun. If you want to feel positive, you got to have energy. If you want to feel alive, you got to have energy. If you want just to have a productive life, you want energy. So energy is a currency here. And time is of the essence, because I think time should not be wasted with mito patients. And that's why we decided just to take a very simple approach, but we're going to test it. It's coming live this fall, so stay tuned.
Yeah. I think there's so much hope for therapies in mitochondrial disease, especially the complex 1 diseases. We're developing a new gene therapy for complex 1 disease. That's again, agnostic of the mutation. Again, it's early and we're in models to test it. There's nutritional therapies that I think have real promise. There's oxidative stress targeted therapies that probably are very important. That's sort of the class where Adebenone was in, but Adebenone is just one. There's like many therapies in the class, and I've seen patients vision get better, on some of the ones that we try. And you can go from try in the first few cases. At some point you want to do a trial and actually show it, because then that becomes the new baseline therapy that's approved by the FDA, that's paid for by insurance, that is well made under regulatory standards and is reliable and contains what you need. And then we've also been developing new tests. Like, for example, a lot of our mito patients have too much oxidative stress. A lot of our complex 1 patients have too much what we call NAD stress, redox stress. It's sort of the fuel for the mitochondria. And we've been able to give some therapies already in clinic that reverse those blood signatures of stress. Now, a study should be done. Is that happening in the LHON families, right? The LHON patients, or is there more stress in the people that lose their vision than in the carriers? Do these therapies prevent, the carriers from converting? Do they reverse the vision problem? So in the beginning, when there's not a voice in a community, you try your best, right, for each patient. But at some point you take the learnings, Dr. Sadun and I work together, and Dr. Trabulsi and I work together, and Dr. Ailman and I work together. And we say, like, what have you noticed? What have you seen? And that's like sort of the initial steps. And then it goes into, well, we really need to test this. And the way to test things are clinical trials at different stages. And that's where the families are so incredibly important. And you're organizing, the information and the natural history and the consortia and the access to samples. And then we can help do everything. We can't do everything. And I always say, I learned this from the families quite a while ago. We can't do things for you. We can do things with you. And that's, I think, a really important lesson that families have taught me. Your voice, sort of your prioritization, you're giving legitimacy to scientists saying, come work on this. This is important. Don't go work in a cancer lab. Come work on an optic neuropathy lab. Come put your brain power behind this. Doctors, focus on this. That's what the advocacy groups do in a really important and meaningful way. If you were at the dinner last night, right, you gave a research award to a young scientist who's probably now going to spend her career in mitochondrial biology, right? So you've just won another win, right? You're bringing more people to work on the problems.
We've talked about therapies a lot, but Marni, you mentioned something that is kind of the golden opportunity that I see for LHON. And actually, Chris and I were speaking with a biotech, and we're talking about, this therapeutic option. He goes, can I ask you a question? Is there a preventative for LHON? And I said, there is not. We still need to better understand LHON. And so this is another thing that happened when gene therapy got a lot of attention and a lot of NIH funding. Look out for a Giving Smarter guide that will come out. It'll tell about the funding landscape as well. But one of the things that happened is we thought that silver bullet would enable us to just say, okay, you have vision loss, we can restore, you're going to be fine. But prevention is such an incredible opportunity. And it's going to require us to better understand LHON from beginning to end, the carriers, the affected, the wild type like Chris. And so I'd love to just get your perspectives on prevention research and maybe even backing up and prevention funding. One of the things I heard on my interviews was the NIH shouldn't be called the National Institutes of Health. They should be called the National Institutes of Disease because they just try and fix diseases. What does it look like to do prevention research? What are the things that we need to start saying we can keep these individuals from converting?
I think that's incredibly important. There's so many, probably more people in your families that are carriers, right, that have converted. And there's a lot of fear, right? Like, what is it that I'm going to do that's going to make me convert? Or was there something I could have done to prevent it from my child, right? There's a lot of guilt. There's a lot of concern. These are very real. I'm going to go back to the meeting that I was in this morning where they developed a gene therapy for this terrible neuromuscular disease where kids don't move, they become paralyzed. They can't pick up their head as babies. And now there's a newborn screen in every single state that everybody's child born in America is screened for this terrible disease. And if you start the therapy within the first 6 weeks, they kind of look normal. That's unbelievable, right? To go from like completely paralyzed to like, and not barely moving their eyes, right? To like riding bikes, right? Like just night and day, right? They don't just have a gene therapy. They have 2 drug therapies too. They have multiple shots on goal and multiple approvals through the FDA. And I was just talking to the researcher who's been leading this, Dr. Finkel, who's now at St. Jude's. And I said, well, why wouldn't you just start everybody on the gene therapy? And he says, well, we learned that some people get antibodies and they can't be on it for a period of time. So, but we need to get them on a different therapy because if we don't start early, then they have irreversible neuronal loss. So the community has worked incredibly well together to say, this and this might go together. This might come first. This might come second. It's back to that arsenal, theory that you need multiple ways, to treat. And in order to get on the newborn screen, like why isn't every baby on Earth screened for the Leber mutations? Have you asked that question? Could they be? There's rules. And the first time they tried to get this other disease, SMA, on, they were told no. But then when there was a treatment, they were told yes. So one of the rules is that you can intervene in order to get babies tested at birth. So then the question is, okay, somebody's affected, has a risk at birth. Then it's like, well, would they have a risk of being affected as a child? Should they be monitored differently, right? Would there be an intervention? And so these are really unbelievable questions, right? If you can prevent somebody from losing their vision, or you can give them life tips or medication, okay, exercise works. Let's mimic exercise, right? Like, what is it we can give you? What is it about exercise? We talk about the mitochondrial cocktail. I hate that phrase. Cocktails are at parties. I think medicines are in the clinic. What is it?
That's fun.
Yeah, they're fun. But I think they also, they undercut the point that they're therapeutic, whether you're getting it from the health food store or from a natural product or from wherever. If you're using it to treat a disease, it's a therapy, right? It's a treatment. And so I think we need to take it very seriously, what's in there. And I think families will tell you they feel different. Affected individuals will feel different. But when do we start them, right? Do you start them at birth? Do you start them at a certain age? Is there a test like based upon your breath or your eye findings or some other symptoms you're having? So I think prevention is the big ticket here. I think in any genetic disease, right? Yes, you have a genetic predisposition, but right, you have a genetic predisposition to cancer. We take that very seriously. If you have a 5% chance of having breast cancer, there's some serious interventions that are offered to you, right? Whether you're a man or a woman. But if you have a 15% chance of losing vision, there is nothing that is offered to you, right? That's like an issue, right? I think that's one that, we can raise our voices and make more clear, right? The community wants preventative knowledge and interventions and therapies. And I think that's going to come, like I said, from the advocacy of the organization.
I think the only way to coerce the NIH study section to pass a study section just to fund the prevention-based studies is to angle it a different ways. And the way to do it, I think, is just by asking the question, what are the genetic modifiers? Because if everyone gets tested and then say that 11778, okay, fine, but doesn't mean that you're going to convert. So if we know the genetic modifiers, so it depends how you notice, and the next session depends how you ask the questions. So maybe not formulated as prevention, but as uncovering and masking what makes the penetrance of the mutation just to go totally 100%. And that's how you can actually go to study prevention, because that's the only way you can convince scientists that it's a worthy question on a human base, but I mean, on a scientific base, how do you prove prevention? Because if you have no symptoms. So I think genetic modifiers and epigenetic as well.
I think if I wanted to simplify for you what I've been doing the last 20 years, I think mitochondrial diseases, especially complex 1 diseases, they're diseases of stress. That's how I understand them. Not having energy, not being able to handle free radicals, these things are very stressful to cells. And so I think some of the theory of prevention is what are the stressors, depending on who you are and your genetic background, that will tip you over the edge from adapting to the stress to succumbing to the stress, right? And so stress could be nutritional. Some people show up when they're fasting for a surgical procedure, right? That's when their vision loss happens. People forget to, you don't forget to feed a kid in the hospital, but an adult goes in the hospital and people forget to feed you, right? Oh, we forgot to give the diet. Well, 2-3 days without eating, that's very stressful, right? On the cells of the body. I'd assume we eat every 3 hours here, right? So I think you understand that. Viruses, right? Very stressful, right? On a lot of people show up after a cold. Mitochondrial biology is very related to viral response, and we're at the beginning of really understanding this. But often not bacterial infections, viral infections are what push people over the edge. Other stresses, trauma, right? That's very stressful. Some people show up at the time of trauma. So I think prevention and knowledge is what are the factors and what are the stressors that allow the cell to go from handling it to not. And I think that we're going to learn a lot. And so, like I said, there's medications that even when you're not making energy, you can quiet the stress in the cell. And I think those are approaches we need to seriously consider, because gene therapy is still a long way away. I mean, decades for every type of genetic disease that we have and every type of cell that we have, right? Like making all of that understanding, it's still not going to be available tomorrow. So in the meantime, there are real interventions to lower stress in your life and in your cells. And I think that is an enormously important area in this condition.
I think one thing that hopefully you're seeing as a common thread throughout here is that we need to understand the mechanism of disease. We need to understand what's happening in the patients, what's happening in the carriers, what's happening at conversion. And so I think that there is an immense opportunity, starting with the resources of patient data and samples, moving into models, and then having a collaborative research ecosystem that is doing the research to understand the biology far better. I know that we have a very limited time. I did want to offer the opportunity for any questions if anyone has them. I believe that there's a microphone that Brooke can grab from Jessica.
Jessica, you got a microphone on your right side. There you go.
If there are any, okay, wonderful. We've already got some hands up.
So now that we're getting our data in, and globally you're going to have our information and our symptoms, do you ever foresee a time when at a mitochondrial conference you could be gathering skin samples from the people that are here that are motivated?
Yes. But it's not just skin. I mean, it's multiple assessments, right? So having everybody here ready to go and figuring out, like, here's the surveys to fill out, here's the assessments we're going to do. So, and then doing it year after year, that's called a prospective history. That is so valuable, so year after year, collecting that data, that's one of the more powerful things a community can do.
Think about like a timeline as a horizontal, right? And being able to actually see how things are changing, which is why when Melinda was talking about the data collection program yesterday, she's like, if you haven't gone back into the DCP registry, please do, because it's both the physical skin sample and other aspects, as well as those qualitative inputs that we think can
right.
That's what the FDA wants. Like, you just nailed it. Like that's what they want.
Yeah.
Yeah, and maybe they will uncover the stressors as well, because you can associate what happened in your life with an event. I mean, that led
Right.
to
You might say,
change.
since last year I've had surgery. Since last year I've been under a lot of emotional stress. I lost somebody. Like whatever the stressors are. If that information is collected in real time and you see, and we do a vision assessment while you're here, or vision assessments, because there's no such thing as one, then that's a sort of thing that, and the community makes it available to any company developing therapeutics, right? That becomes a very rich resource that actually attracts more companies to do therapeutics, because this information is there and available to them.
Sometimes we toss out big lines like your voice matters. But I hope you're hearing like there's very really concrete ways that we can create structured data around your voice and your input. Brooke, you had another question?
Hi. So 3 of my 4 my kids have lost their vision. So the fourth, she's wondering if there are things she can do, and you mentioned, well, there are antioxidants or diet. Are there specific things she should be taking? I know vitamin D has been mentioned. I think it's called quinol has been mentioned. Someone said one person said, yes, you should start an idebenone even if you're asymptomatic. And another person says, no, you should not do that. Just take QAnon. So are there recommendations that you have?
It's such a good question. And I think it's more experience-based than having real guidelines based. So I've been facing this question for quite some time because I see a lot of families and siblings of kids. And so I think there's this concept in medicine called equipoise, right? Like we're not sure, right? But what's the relative risk and the relative balance? So we've published articles and we can share them with you about like what therapy should be considered in anybody whose mitochondrial function is impaired by a genetic problem, right? And so we do some tests where we look in the blood at glutathione. We look at this thing called redox balance. So we look to see if we see signatures of the cells under stress in your body, right? We could look at coenzyme Q levels, not in the blood because not that reliable. CoQ is very fatty. And so just in the blood alone, it's not as informative as we look inside the white blood cells. It's called leukocyte CoQ. So we can look for signs of deficiency, but at the end of the day, we're not looking in the eye, right? We don't have a way to measure it in the eye. So what we err on is a side of, well, here's a combination of medicines like multivitamin, right? B50 vitamin that kind of a lot of cofactors for how the mitochondria are working. Not everybody has an ideal diet, right? So just giving your body like enough of the routine cofactors seems like a good idea, right? So complex 1, for example, needs riboflavin, B2, right? And so, but some B2 can make people nauseous. So we have to give the right dose where they can tolerate it, and we have to step it up. Again, I've never seen a head-to-head comparison of like different antioxidants, vitamin E versus lipoic acid versus N-acetylcysteine versus CoQ. But what we know is that these are very well tolerated. There's very low risk. There could be side effects, but they're typically not. And so when we see families, we say, well, why don't we do the things that we know are well tolerated? So we'll often put somebody on like vitamin E, which is a general antioxidant. We call it like a paper towel, kind of cleaning up the free radicals. It's never going to be effective. You're always going to make more. It's like a waterfall, right? But it's doing something. And then we use CoQ, and then N-acetylcysteine as well as a very nice brain and neuronal penetrant amino acid, like N-acetylcysteine. We call it NAC, NAC. It gets into the neurons and it helps the body fight off free radicals. So it itself is not an antioxidant, but it helps the body boost its major antioxidant response. That said, there's no proof that any of these, will or won't work, but it's the equipoise. These are things that are very well tolerated that you can be doing that help your body, fight stress, right? And so that's the whole idea. It makes you a little more resilient. So will you feel different? Some people say they do, but for the most part, the idea is to give your body more support in just being resilient to stress. And so have we seen people, vision, even after they've been affected, return? Yes. Does that happen naturally in LHON sometimes? Yes. So are we sure? No. But do we know that they work any better or worse than idebenone? We don't. And idebenone in America, at least, has had a problem of access. Not everybody can get it. And then you're right. The ophthalmologists argue about whether we should be giving it and whether maybe in the 3460 mutation we shouldn't be using it, but in another mutation we should. And when do we use it? So I agree. There's a lot this community needs to do to figure this out and can be done. But I think in the meantime, if there are things that are very well tolerated and you can try them and see, you could work with your doctor. There's some very safe levels of these medications you could use. And they're at least, if you would like to try, something, these are things that we use in our clinical practice.
So clearly, if you were a stenographer, you were capturing a heck of a lot more on the question side than on the answer side of the equation today. But the reality is we need these questions. We need these really wonderful insights to inform our prioritized path forward. I promised, Caitlyn and I promised Maria and Jessica we would finish on time at 11:45. So we're going to wrap it there. We'll come back for questions at the next break if that's okay. Caitlyn, you want to close us out?
I would very much like to close this out. Thank you all so much for your attention. I would love to have any questions. You can get my card. We can have a conversation if you'd like to learn more about the process. We will be in collaboration with the LHON Collective, putting out a Giving Smarter guide that will outline the opportunities that we've learned about, that will outline the funding landscapes. You can learn more about some of our findings during this. You probably saw that there was an executive summary of our state of the science. You can look at that. I can share the executive science, share the state of the science. If you have anything you'd like to talk about, please reach out to those of us on the stage. We're really happy to connect with you. Thank you all.
Would you please join me with a round of applause? Thank you, ladies. Thank you, Doctor.